Neoscience Hub. As we all know, science impacts every aspect of our lives. But rarely do we pause and think about the whys and hows of this. Neoscience Hub attempts to contribute towards this connect between people, science and technology. Today in our interaction session, we have a distinguished scientist, Dr. Rakesh Mishra, ex-director of CCMB, Center for Cellular and Molecular Biology. Sir has been director for CCMB from 2016 to 2021. Dr. Mishra has been recognized for his research and achievements and has been elected by many respected bodies such as Indian Academy of Sciences, National Academy of Sciences and Andhra Pradesh Academy of Sciences. We are privileged to have Sir chatting with us. Neoscience Hub welcomes you, Sir. Sir, can you elaborate on your research works?
So my research interests are how information is encoded in the genome, which is the genetic material for all living beings. And you can appreciate that in all our cells in the body, they have identical genetic material. We have about 50 trillion cells that to make a human being. And each cell has identical genetic material which we inherit from our parents. But still, the cell in the brain work for functioning, thinking and feelings. The cells in the stomach and intestine work for digesting food. Cells in the liver for do detoxifying and many other functions. Cells in muscle work for running. So how the same information is used in different cells so that we function like an organism is the basic question that my lab is interested in. We call it developmental biology. All the organisms begin their life from a single egg, which get fertilized and then eventually becomes an individual who can walk, run, fly, depending upon which species the egg belongs to. And one of the approach that we take is that there are genes in our genome, but there are other than genes, much of our genome, 98% of our genome doesn't have genes that code for protein. It's something else, and that's what we are trying to figure out. What is it else that makes organisms very special? And one of the way we propose is that each cell type or tissue type, the same genetic material is packaged in different ways so that in brain cells, the enzymes that digest food are not traceable. They are hidden inside and so that they are not used. So brain doesn't have to produce enzyme to digest protein, otherwise it will digest itself. While the cells in the stomach have that enzyme gene available so that they can digest proteins, so we survive because otherwise we'll starve. So this, what are this packaging code in the genome is what we are interested in the lab. And the model system we use for our experimentation are the small fly called Drosophila or fruit fly and a small fish called zebrafish. So fish as a vertebrate model and fly as an invertebrate or insect model. And we have learned enough that we can actually change the shape and size of the fly or a fish. We can make the fly longer, shorter, make the fly with 4 wings, no wings, or other changes. So it helps us understand, and these mechanisms that operate in the flies or fish, they also operate essentially in human beings also. So once we understand how our body is formed, then something goes wrong in our body, we can try to fix that so we can have a healthier life. And many other questions that why we live 70 years, 80 years, 90 years and not 500 years. Why other animals live few years, like mouse lives 3 or 4 years. In the same, dogs live 15, 16 years, 18 years, like that. So there are many other questions related that are all embedded in our genome, but we don't know how to read that. And that's the interest that we have in the lab, which we do by experiment, essentially understanding the genome and its functions.
What are your achievements, sir?
So as a director, my achievements are of different kind. Then the whole lab has to deliver like my own personal research interest is what I just talked to you about, in which we understood some parts of genome for which we did not know the function, but we know they exist. Now we know that they have function, and we know more specialized way, more specific way what kind of function these sequence elements do. We call them simple sequence repeats. They make about 2% of our genome, but they are even larger percent in many organisms. And we know that they are involved in packaging and making independent domains of the functional domains in the genome so that genes can have their own regulatory environment and not get influenced by neighboring genes. And that's how the complex organisms evolve. CCMB as an institution, when I was heading CCMB for about 5 years, we have CCMB is a research laboratory that is involved in addressing fundamental questions of biology. And we realize that while we are doing very well in fundamental biology and that has to be supported, there are also findings or discoveries that can be useful for the people. In the past, CCMB has done remarkably well in establishing the technique like DNA fingerprinting, which is used to identify individuals today, and later developed technology to identify even organism, any animal. We can, if you give a little piece of hair or blood or flesh, we can tell which animal it is. And CCMB also used its basic understanding of systems to make new varieties of rice, which is very popular and which is now given to farmer for many years and grown in several states. What we did in past few years was to organize these activities and institutionalize the advantages or discoveries that we get in the lab, or entrepreneurs who have such ideas and they want to try out their ideas in the labs of highest quality. We created innovation center, a whole dedicated building for this, which we got support from Niti Aayog. Is called Atal Incubation Center, which is one of the first a government lab could set up in a biomedical research area. And we have been then interacting and helping large number of startups and innovators, young people, even our own PhD students, when they are interested in doing some applied research, take some discoveries for the people, they can come and use that space for their ideas and deliver their findings and discoveries to societal use. And of course, we made many other, our colleagues made many fundamental discoveries during these several years of how cellular processes are regulated, how cells divide in bacteria, new enzymes and new factors in that which may give us a target for new antibiotics. As you know, the antibiotics are going to become useless because the microbes are developing resistance against them. So we have to find new targets, and these kinds of discoveries go a long way in addressing the problems like AMR and so on. So like that, many other discoveries and sophisticated facilities of very highest quality like cryo-EM and so on, those kinds of things. They help us solving the challenging problems. Very strong sequencing, genome sequencing capacity we created, made it multifold in capacity, and that has helped a lot in addressing the genome surveillance of the COVID when it became a problem. And because of our entrepreneurial preparedness and facilities, we were able to very quickly culture the virals, SARS-CoV-2 in the lab, and help many labs to test, companies to test new compounds, new drugs, new sterilizing agents. And even we developed a very simple and cheap and fast RT-PCR method to detect the virus called dry swab method, which helps a lot in reducing the time and increasing the throughput of the existing method, which has been now approved by regulatory agencies, and many companies are using this technology. So, being a fundamental research laboratory, if we are prepared and we are open to innovations and new ideas, open to interaction with industry, we can contribute significantly, which is what we have shown during the pandemic, that even being a basic biology lab, we responded to the problems of the society and made several contributions for which we can be proud of.
Sir, you have been working on mRNA vaccine. Can you explain us on the difference between mRNA and traditional vaccine?
Also, mRNA vaccine. So, I can your first question first, that we have began the setting of the technology platform for the mRNA vaccine, and there are 3-4 components of that. One is making the mRNA, then purifying the mRNA when it is in a stable and active form, and then packaging them into a lipid particles, which is very special. And then once that is done, then third is to go to animals or inject for testing their functionalities. So the first step we have established, certainly and then now we are in the process of packaging the mRNA in the what is called LNP, the lipid nanoparticles, and then we will be testing them in the animals. So, and these are very disease agnostic platform, so it can be against any infectious agent for we want to make vaccine. Even it can be used for making other therapeutics where proteins are required or peptides are required. They can be used as therapeutics, not only for vaccines. Coming to your second questions, second question, how different the mRNA vaccine is compared to other vaccines. So normally vaccine means the organism which is troubling us, you kill that organism. So either a whole organism or a part of it you inject in the body, which is not going to cause the disease because it's killed by heat or some other chemical means, or only a pinch of that parasite is taken, pathogen is taken, and that protein or set of proteins, and they are given to the body. So body will find the new agent and make a defense system against it by triggering the immune system. We have adaptive immune system, which any foreign agent comes and our body prepares in 2-3 weeks, so that it can prevent any further infection or any damage by that pathogen. So a dead pathogen or a component or some parts of the pathogen give the same advantage to the body. It gets, it remembers that this kind of thing has come and it will inactivate it. But what happens in mRNA is, as you know, all the proteins are made from the genomic material, which is generally DNA. Sometimes it is RNA, like in the current SARS-CoV-2, it is RNA, which is a genetic material. It doesn't go through the DNA avatar of it. So which means that if we have, and mRNA means messenger RNA. So the RNAs are of many kind. When gene gets transcribed, converted part of that genome into RNA, and which is ready to go for translation to make protein, is called mRNA, is messenger RNA. So what we do in the lab is to make a template in the bacteria, and by transcription in test tubes, we make lots and lots of RNA or mRNA, you can call. This mRNA is special because it is modified in some ways that while it has the same message, that if it translated, it will convert the protein for which it has been designed. But it has been modified sufficiently that it is very stable, and it does not create a immediate immune response against the RNA itself, because RNA is not the target. We want the protein to be recognized by the immune system. And so and then this mRNA is packaged into little bubbles of oil and injected in the body. So when it goes into the body, the cells take this little bubble nanoparticles and put the mRNA into the cytoplasm of the cells. And cytoplasm, purpose of the cytoplasm is to translate the mRNA. So now this mRNA will be translated into proteins. If it is a SARS-CoV-2 genomic component which we have converted into this mRNA, then it will make SARS-CoV-2 protein in the cell. So let's say you take the spike protein gene make the mRNA and put in the body in the method I just described. So it will make now spike protein in the body. This spike protein will be exactly the way spike protein is made in the body in normal infection, because you have given a messenger in the cell. Now messenger is being read, being translated, being modified. Most proteins after being translated, they get modified in many different ways. Chemical moieties are added on them, which is called post-translational modification. They are functionally very, very important. And since this is being made in the human cells in the body as the which is also faces the infection, so it will make exactly the same kind of protein. Therefore, the immune response will be very accurate. It is not that protein that is made in bacteria or yeast or any cell culture and then injected in the body. It is the antigen prepared by your own body. So you get your own immune response very effectively. And one mRNA molecule will not make only one time. It can make many, many times, hundreds of times the same protein, because it's translated again and again. That's what the system does in the body. So which means very little amount of mRNA can make lots of proteins in the body, and we can achieve the vaccination level of protein easily. The other advantage of mRNA vaccine is that it's very easy actually to make. Once you know the genomic sequence of the organism, it just takes a couple of weeks to make the template in large quantity, translate, and then you can start testing and trying. And in 3-4 months, actually, you can go for human trials or usage, depending upon where you are. So it's very fast, and it can, for example, you can make now a vaccine against Omicron. Earlier vaccine is against Wuhan variant. Then you can make vaccine against Delta variant in a very short period of time. So it's a revolution actually in the field of vaccine and in the therapeutics. It's going to be cheap, it is going to be more effective, and it's going to be very, very efficient because very small amount of the mRNA is required. One of the Indian companies in Pune, Genova Biotherapeutics Company, is making a version of mRNA vaccine where it's a self-replicating mRNA. So that means when you put in the body, it will make many copies of its own. Then it will get translated. So which means you need even little material to put in the body, very minute quantity. So there is no side effect and there is no other things. It will make lots of RNA, and then it will even more amount of protein, and the formulations are very stable. I think it's final stages of trials, and very soon we may get the news that is going to be accepted by further vaccination programs.
What are the different types of COVID vaccines available in India? And how does a common man know which one is good for him?
So I think in India, most people are vaccinated by Covishield, and maybe 10-15% are vaccinated by Covaxin. So we didn't have much choice at that time. Everybody was given Covishield, which is a fantastic vaccine, and so is Covaxin. I'm not sure if Sputnik is still available publicly. They may have been given as a trial and those things. So, but they will be available. So there are many different kind, and all these vaccines are good. You see, it's like principles used by most vaccines are of very general kind. There are 3-4 different ways of making vaccine, and they fall in one of these categories. You have mRNA vaccine we talked. Then there's vaccine like Covishield, which uses another viral vector, and then vaccine like Covaxin, which just make the virus that you want, kill it, and then inject in the people in a simple ways. So there are some DNA-based vaccines. They essentially all of them give the same spike protein to the immune system, and that spike protein is used as an antigen, and the immune system activates and launches protection against that. So they all are very good actually, and they have been tested, and then only they are approved. So there is actually no big difference in these vaccines. Some people consider mRNA vaccines as more efficient because they are you are making the antigen in the body fresh and doing, and you can also make them very fast for the future variant specific and so on. But we are yet to see the long-term effects of thing, whether memory is more in the mRNA vaccine or things like Covaxin, where entire virus has been given as a vaccine. So, which means some portion of the virus will change and some will not change in new variants. So that should be even more robust compared to others. But at the same time, when you are giving mRNA vaccine, the whole antigen is provided in a very native version, and it will read it like as if coming fresh from the virus and protect us from there. So different things. This is early days for these vaccines, and they are all very good. And I think any vaccine you take, it should do the job. Also, please remember that vaccine is one component. Your body, vaccine is like a trigger to your body to create a defense system. If we are very old, or if we have some comorbidity, our immune system is compromised in some way, any vaccine will be not very effective. It will be less effective, and it might require booster more frequent than others, and so on. So there are many other things that come into play. But by and large, any vaccine that is made available has a backing data that these are safe for sure, all of them, and their efficiency may be 70%, 80%, 90%, things like that, which is in a range. It's small. I mean, these differences are there, but anything WHO says more than 50% protection is very good one, and you can go for that. But these are far more than that. And if everybody has been vaccinated and the doses are available, and there is this, without stressing the our healthcare system, if we can give a precautionary dose or a booster dose, that's a good thing to have. It gives a little extra protection. But I think simple two-dose vaccination is pretty good. We can be confident that we can fight the pandemic very well, most of us. So these vaccines have been tested for disease severity, and they have not been designed or tested for infection. So infection will take place. That's what have been said from the beginning. And the virus is the biggest strength of this virus. In fact, many pathogens is that they can change their shape and mutate very fast. So next time when it comes, body is prepared for the old virus that it was injected with, but new virus look little bit different. By the time body realizes, oh, this is the enemy, it has already entered and started doing some job. But by then body will recover, will realize that this is the same nasty fellow, so we have to catch it, and then it will stop the spread of the disease. So there are, if the vaccination has been recent, or a booster was given, and there is a lot of antibody circulating in the blood, the any moment virus comes, antibody will go and neutralize the virus, and virus will not be able to infect or infect very little. But if the vaccine is 6 months, 8 months, the second dose was that much old, or some people lose antibody titers sooner, then virus will be able to infect there, because antibody is not immediately available. But when the cells fall sick, the cellular immunity comes. There are cells in our body that read if a cell is sick, they kill the cell and remove from the system. So when they realize that themselves are infected with the virus, then they go and attack those cells and make us recover, get rid of the virus. That is why disease doesn't go very far. Only few cells get infected, but we can, by then our system takes care of that. So that's why vaccines are very much, even they may not be able to stop the infection, but they will certainly stop expression of the disease, or setting up of the disease and the clinical symptoms. So certainly that will be very, very useful. And as you can see, when we are going through the third wave, I mean, 3 lakh people, 3.5 lakh people every day getting infected. But there is no panic in the hospitals. There is no hospitals, even compared to the second wave, maybe the hospitalization is
Less.
20%, 25% of that. Not only less, is very significantly less. ICUs are one third, one fourth compared to what was in the second wave. So this is because of, well, largely because of vaccine, and also we should not forget that very huge number of us got infected during the second wave. The sero surveillance data shows that 70-80% of the Indians got infected during the second wave. In the cities, many cities, big cities, even more than 90%. So that memory is also there, and then vaccine has been given over and above that. So we are pretty very strong position in the virus. It can come and infect, but will not be able to do much to us. So if we so the vaccine has helped a lot in controlling the damage. Otherwise, even if it is mild virus, it can cause more damage. And most of the time, what data suggests is that the people who end up in hospitals or in ICUs are the people either they have some other immune problem or comorbidity, or most of the time they are not vaccinated people. That's why they have ended up there. So that has helped. The vaccines have worked really well.
Sir, are multiple jabs our only way out? Are we anywhere near a single shot for lifelong immunity against this infectious disease?
It's very it's risky to predict that we will have a single jab vaccine. Maybe we will have. There are many labs and companies with the help of certain governments. They are working not in India but in America and in Europe. There are companies that are working, and they are considering the bringing in so many variations and combinations in the single jab that it will protect against. Not only all variants, it will protect against all coronaviruses. Their dream is very big, but so it can happen, but it will take time. I mean, it's only uh, these are things only when we do you can tell whether it's working or not. Theoretically, it is possible, but practically, it will be possible or not. We will know in coming several months or a couple of years. Now people know that vaccines are so important for such pandemic kind of thing, and pandemics are not, this is not the first or last pandemic. More will come because we are such a it's one global village, right? People by the time you realize there's Omicron in South Africa, it's already there in dozens of countries. So it's that much if travel and interaction is there. Pandemics will happen. So we have to be prepared. So preparedness is largely by vaccine. Anything else, any drug will work after you fall sick, but vaccine will let you not even fall sick, right? So preparedness is that. So, a single dose vaccine for long-term protection is a possibility, and I'm sure the amount of in this pandemic, many good things have happened. One is uh, vaccines have got approval very fast. No vaccine was given approval with this short trials. So that means, and which has saved lives. So that means now regulatory bodies know that once the safety is established, we have to give more chance for a drug or a medicine or a vaccine to succeed. So if it is not harmful, we should be more accommodative and be more little bit more adventurous. And with the techniques like mRNA and also we can be very quick response time. In fact, from the day genome was announced, within I think 3 or 4 months, Pfizer and Moderna were ready with the vaccine for trials. So which means it's very fast. So those are things that are going to be very much there for everybody to use and will help us. But at the moment, it looks like these vaccines they give long-term protection, but antibody titer goes down. So maybe after 6 months or 8 months, if you take a booster, you will be even better protected. But that is not necessary, and maybe people with the more difficulties, like very senior people or with comorbidities or some other problems, or people who are going and fighting against this virus, like frontline healthcare workers, they should be given precautionary dose, but others don't need. For others, we have other way of managing this disease. We don't have to uh, vaccinate ourselves and then feel that no, nothing can do anything and I can go and roam around. The best vaccine you have is the mask. If you wear the mask, then no virus can come. Any variant, any kind, it can never penetrate your mask. So if we do that, so I hope that we have taken this lesson and we make it part of our civilization and respect to others, that we wear mask when we are in public, so that we are not helping the virus using me as a vehicle and spreading around, or I may be unknowingly infected and I'm not spreading to others uh, an infection. So that should be taken as it's like we say that you should not smoke in front of others on their mouth, you should not spit in front of others. Similarly, if you are not wearing the mask, you are smoking or spitting virus on others. So we should not do that. Now, since we know it, we should not be doing that. That's what I meant by being cultured or civil by using these methods. So we should, if we learn that way, the way we wear glasses, the way we wear the cap, the way we wear shirt, and we lot of jewelry people wear. Same way we should wear mask, maybe make it nice one, more designed ones, and so on. And that should become part of our culture, particularly when we are going to a crowded space or unknown crowd or closed space. That will help. So virus will be choked. If it infects also, less virus infects me, but I'm wearing mask so well that is not going to get out. So virus will, in 2 weeks, it's over. So virus, I have killed the virus and not let it to go and play with my other family members. So that is other part we have to learn and do. That will go a long way. That will actually kill the pandemic much more efficiently, because it works instantaneously. It doesn't have to worry about whether you have immune compromise, comorbidity, or 2 weeks to vaccine to set in and all. The moment you wear properly mask, you are set. So those are things, and the vaccine, of course, and the seropositivity. I see that now we will be out of this difficult time in coming months.
Considering that we have many brands in our Indian market, is mix and match a safe option?
These are things, there is no easy way of saying yes or no to it. These are things that should come by trial and experimental process. So unless we try various things, if you have, when you have 5, 6 vaccines, if you consider permutation combination, it becomes so many. So now which will be first Covaxin, then Covishield, then this, then that, then mRNA? How you decide which one is good or which one is not? One thing is for sure, that they are very safe, because that has been established very well. So even if you take the third dose of some other vaccine, in terms of safety, I think is going to be no problem, because these have been tested and they are very safe vaccines. Now, whether it will give better protection compared to the same vaccine which was given is anybody's guess. It's not easy to tell. Some people will say, yeah, now you are giving new vaccines, so you are increasing the repertoire, you are increasing the antibody varieties and so on. But the first one, if it works and all the antibodies are there in big way, that also provides good protection. So the very simple answer to your question is that since we do not know these things, which combination is good, the best is to take the same vaccine as a third dose. So, and which will do as well. Maybe you get 15% extra protection, and some other permutation combination will give 5% more or 2% more. Doesn't really make a big difference in practical life. The till we know that a combination, certain mix and match is working better than other, it is best to take the same vaccine as and when it becomes available as a precautionary dose. Later, if let's say we have taken Covaxin or Covishield and mRNA is tried and that gives a good result, so why not? But that at the moment is only idea. We do not know that whether it will be useful or it may not work at all, or work very little. So, but we know that if we take the third dose of the same vaccine, it will be certainly useful. So we have to wait till the mix and match results are available. People are trying that. Like in India, I know the studies are going on. If you've taken Covaxin, give a booster of Covishield and vice versa, and see if you get the same result or better results. Same vaccine, two dose, or first Covaxin, then Covishield, or first Covishield, then Covaxin. These three things we will know in coming 2 3 months. But other things will still require more studies and so on. So we will get to know, but I think we can be very confident that just take the third dose of the same vaccine and you will be doing as well. So it shouldn't be a problem, but we should not try many experimentation till proper studies have been done. Let's not use our innovative ideas and now I will try this vaccine. It's not a restaurant to eat another vegetable or another non-different kind. These are very well tried and tested, and we should respect that study.
Where does our country stand in terms of vaccine cutting edge research for future generations?
See, India is now uh, technology ready for most, I will not say any kind, but there, I can't remember something in biology which we can't do here, and we have to go somewhere else for that research. When we began our career, there were no internet, there were no emails, there were very little use of computers. We have to wait for several months for a journal to come to India so that we can read what has been published and so on. And similarly, there were very few labs which have facilities, and we did not know the latest technologies and all. Now anything is published. If Nature and Science are published Thursday in UK or US, the same day we also get it. Our students also read the Nature the same day as Americans or Europeans read. So information technology has really brought the revolution that we are at par in terms of current literature. I will thank COVID for bringing this virtual conference mode like we are talking. So now we are having lots of meetings and discussion internationally without having to travel. So that has improved lots of, and I attended some terrific meetings which you have to make 1 week of travel, visa and all, then to go for that. Here you can do without any travel. And in terms of infrastructure and facilities, we have a number of labs which are second to none, like laboratories like CCMB. Even if you go outside anywhere in the world, you will have to search for such a sophisticated and well technology enabled labs. You will not find many. Most labs will not have so many facilities under one roof and available to the staff and scientists with such a free scale. And we have other advantage of very young bright people. So now if we can, and that is our biggest asset, not the machines can come, they will keep changing, they will keep getting outdated, but our people now don't have to go to America and Europe to do the cutting edge research. They can do right here, and that's what is going to be the game changer. Machine and all, of course, the facilities, other things required. I very strongly feel that the government, center, and also to some extent the states, they should give more funding to the research, because that is what is going to decide how soon and how strong developed country we are going to be. Will not be a developed country or a strong country by buying everything from outside, because you will get material which they want to sell. You will not get material what you want. If you are making your own, you are developing technology for yourself. There are certain diseases which are our own unique disease. If we don't make on our own, we don't develop the product here, we'll have to pay 20 times, 50 times, 100 times the cost. I mean, just give simple example. When this COVID came, the test of COVID kits were such a scarce commodity. 4,000, 5,000 was the cost for one test. Today, you can CCMB and some other laboratories and companies and CCAM, with the help of some very generous funding, we have now endogenized the entire COVID testing kit, and it costs less than 100 rupees. Of course, some profit, something has to be added, but now test can be done in couple of 100 rupees with same quality as was costing us 2 years back 4,000. You can imagine what was the loss and what difference it makes when you make things on your own. So the technology is there. There's no company from abroad or no foreign scientists have come to help us indigenizing all those things. That means everything is there. The capacity is there. The intellectual people are around. Technology is there. Our production capacity there. Everything is now in place. And I think this pandemic has woken up many people that we have to utilize endogenous resources, and then only we'll be able to develop as a strong country and take care of our people. Otherwise, very privileged one only will get the benefits and others will be deprived. And we cannot imagine a society where a lot of people are deprived. That means you are not a developed country at all. You have to, the whole, everybody has to get the benefit of the technology, and their basic needs of medicine or food are met.
Thank you, sir, for enlightening us from vaccination approval to preparedness of vaccine and possibility of single shot for this infectious disease. We also agree with your thoughts on how crucial it is to mask up and protect ourselves. Thank you, sir.